Patients are screened in 18 sites in 15 countries all around Europe. Patients are selected based on the main inclusion criteria, which is a diagnosis of lung cancer, malignant pleural mesothelioma of a thymic malignancy, any stage at any time of their treatment phase. The requirement is the availability of tumor tissue, which is collected and centralized at Gustave Roussy Cancer Campus, where the DNA extraction, the quality control will be performed, and thereafter the DNA will be sent to 14mg, which is based in Cambridge, UK. The objective is to determine the molecular profile of the tumor of these patients, and thereafter, if possible, to offer them the participation to a downstream clinical trial. The method is a next-generation sequencing, so a panel of about 360 to 400 genes will be the one tested for all these patients. All the information will be centralized, so they will be collected through electronic CRFs that are then centralized at the EORTC headquarters in Brussels. The main goal is ideally to screen these patients and to offer them a targeted treatment based on their molecular profile, and of course to assess the quality of the testing performed. The study has been activated in 2 countries, Italy and Slovenia, and so far we have 8 patients enrolled. 500 for the first year and 500 to 1,000 for the following years. Basically, it's important that the analysis is performed centrally, so that the quality assurance is centrally done in Gustave Roussy, and that the testing is performed by 14mg, which is a spin-off of Sanger Institute, so the quality of the analysis ensured likewise.
SPECTA is a program that has 5 branches, one of them being for patients with any thoracic malignancies. The idea comes from the lung cancer. Today we treat roughly 15% of the patients in the daily practice based on the molecular profile, mainly EGFR and ALK. But we know that much more patients could be potentially treated based on the molecular profile, but with very small number of patients per molecular abnormality. So when, as a treating physician, I have a new trial, I have to open one trial for 2 or 3 patients per year. The goal of SPECTA is to have one screening per all, meaning I screen my patient with one large molecular portrait, and based on that, my patient will have I will have a clear figure on what is the molecular abnormality that I can target, and then I will put my patient in clinical trial. But I will not screen each patient in each clinical trial. That is why I have to do today. The concept is one molecular portrait per all the patients. And the idea is not to screen each patient in each trial, but have one screening, and then each clinical trial is plugged into SPECTAlung. So it saves time, and mostly it saves tissue, because in thoracic malignancies, usually we have very small biopsies, so we cannot screen for a lot of different trials. It's very ambitious and it's quite expensive. So it's a huge effort from EORTC, and it's a trial that is run with in collaboration with ETOP. But it's a huge effort and financial effort to support that. And today it's only charity that allowed us to start this program. Well, first, I hope that patients will be treated in a better way and that they will receive more targeted treatment. And then we will have a quite good assessment of what is the molecular epidemiology of thoracic malignancies in Europe, because in 4 to 5 years we should have at least 4,000 molecular profiles of patients with thoracic malignancies.